Senior High School On-Air Academy. Unstoppable! Delivering you quality education amidst pandemic.
To defer school opening to October 5.
Unstoppable!
Giving you accessible learning platform anytime, anywhere.
Ng klase, whatever form it is.
Comprehensible!
Buckle up and get yourself ready.
I will repeat, then you will do it later on.
Accurate!
The sphere that includes all living things.
Today, we will be talking about rational functions.
Simplify.
This is Joss speaking. How may I help you?
This is Ed School Radio's Senior High School On-Air
On-Air
Academy. And now, here's your new episode for Earth Terrific Saya.
Your guide in the subject Earth Science. Earth Terrific Saya.
Earth Terrific Saya.
An amazing day to all of you, our dear learners. I am your radio teacher, Ma'am Johanna R. Caro. May you always have the drive and energy to discover the wonders of Earth and life science. How are you feeling today, learners? Are you excited on our lesson today? Oh, yeah! That's good to hear. I hope that everyone is ready. Make sure that you are at your most convenient place for you to fully listen to our on-air discussion. Before we begin, be sure that you have your student kits with you. Bring out your pen, your paper or notebook, and your learning activity sheet in Earth and Life Science. Let us first check if you have remembered our lesson that we discussed in our last episode. How do animals reproduce? That's correct! Animals can reproduce through sexual reproduction and asexual reproduction. What type of reproduction involves the combination of reproductive cells from two individuals to generate genetically unique offspring? Very good. It's asexual reproduction. How about a reproduction that produces offspring that are genetically identical to the parent? Correct. It's an asexual reproduction. We also discussed different methods of asexual reproduction. What are those methods? Very good. These are budding, fission, fragmentation, and parthenogenesis. How about the two main types of sexual reproduction? Very good. These are hermaphroditism and fertilization. You really still remember your past lesson. Good job! Let's now check your answers from the last episode's quiz. Are you excited to know your score? If so, I want you to set your eyes on your answers and let's start checking. Number 1. It is a type of reproduction where the offspring is genetically identical to the parent. The answer is letter A, asexual. Number 2. This type of fertilization releases eggs and sperm in the environment. The answer is letter B, external. Number 3. A type of asexual reproduction that results from the outgrowth of a part of a cell or body region. The answer is letter A, budding. Number 4. A type of hermaphroditism where the organism is born as a male, then eventually changes to female. The answer is letter A, protandry. Number 5. This process enables and ensures the continuity of species from generation to generation. The answer is letter C, reproduction. Who among you got a perfect score? You did a great job! For those who didn't get a perfect score, don't lose hope. You can do your best next time. Are you still up? Just stay tuned and we will be back for the discussion of our topic.
Mare, narinig mo na ba ang tungkol sa online distance learning ng DepEd? Ano ba ito? Ay oo, Mare. Ang online distance learning ay isang pamamaraan kung saan maihahatid ng mga guro ang kaalaman para sa mga anak natin sa tulong ng teknolohiya gaya ng internet, laptop, computer, at cellphone. Ganun ba, Mare? Oo, Mare. Hindi magkakasama at magkakaharap ang mga estudyante at mga guro. Maaaring matuto ang mga bata sa pamamagitan ng online na pagkatuto. Isang mensahe mula sa kagawaran ng edukasyon at ng himpilang ito.
Good morning! Our topic for today is all about genetic engineering. On this episode, you are expected to describe the process of genetic engineering and evaluate the benefits and risks of using GMOs. Are you excited? If so, let us begin our intellectual journey. As you have learned in our last episode, reproduction is a characteristic of living things essential for survival. Organisms produce offspring which resemble their parents due to transmission of their genetic makeup. The genetic material present in sex cells is used as a carrier of genetic information passed on from parent to offspring through a genetic material called deoxyribonucleic acid or DNA. DNA is the basic hereditary unit in all organisms. It carries the instructions for assembling a variety of proteins responsible for forming a variety of structures. It is a large biomolecule that contains the complete genetic information for an organism. Every cell of living organisms and many viruses contains DNA. The basic building block of a DNA molecule is called a nucleotide, and a single strand of DNA may contain billions of nucleotides. Although each DNA molecule contains many of these building blocks, only 4 unique nucleotides are used to create the entire DNA sequence. These are written as A for adenine, G for guanine, C for cytosine, and T for thymine. Analogous to how the 26 letters of the alphabet can be arranged to create words with different meanings, these 4 nucleotides can be rearranged in sequence to spell the genetic instructions to create all of the different proteins that organisms need to live. Because DNA contains instructions for an organism to create several proteins, it is useful to define another subunit of DNA called genes. Each gene is a small segment of DNA that contains a set of instructions for an organism to create a single protein. A single organism may have thousands of different genes. Together, the entire set of genes for an organism is called its genome. Let's use this analogy to better understand it. Think of the genome as an entire cookbook for an organism. and each gene is an individual recipe in that cookbook. When a single recipe is followed, the result is a specific protein. Why are proteins important? Proteins perform all of the work in organisms. Some functions of proteins include serving as catalysts for reactions, performing cell signaling, transporting molecules across membranes, and creating structures. When a protein is created by its gene, it is said that the gene is expressed or used. Most gene expressions do not produce results visible to the unaided eye. However, some genes, such as those that code for proteins responsible for pigment, do have visual expression. The expression of a gene in an observable manner is called a phenotypic trait. One example is an organism's hair color. In fact, everything you can see in an organism is a result of proteins or protein actions. In practice, since DNA contains the genes to build certain proteins, by changing the DNA sequence, engineers are able to provide a new gene for a cell or organism to create a different protein. The new instructions may supplement the old instructions, such that an extra trait is exhibited, or they may completely replace the old instructions such that a trait is changed. The process of using plants or animals with desirable traits to reproduce offspring with specific traits is known as selective breeding. Selective breeding has been practiced since prehistoric times. Hybridization, or the process of crossing plants or animals with different variations of the same trait to create an organism with the best trait, has been around for many years as well. However, these processes require a long period of time for results to be observed, and usually it is successful only within a close family of species. In the early 1970s, biologists experimented with manipulating and deliberately recombining the DNA molecules of different species. And today, we are going to discuss about genetic engineering. Have you ever seen glow-in-the-dark mice? Are you familiar with the products of genetic engineering? What is genetic engineering? Good guess! It is the process of modifying genes in a living organism to produce genetically modified organisms, also known as transgenic organisms. This method is more rapid and specific than the selective breeding because a gene coding a specific trait is transferred to an organism. Genetic engineering can be done with plants, animals, or bacteria, and other very small organisms. Genetic engineering allows scientists to move desired genes from one plant or animal into another. Genes can also be moved from an animal to a plant or vice versa. How is DNA used in genetic engineering? Changes in the DNA of an organism can cause a change in traits, and its manipulation could lead to the formation of genetically modified organisms. As the knowledge of the DNA advanced, more genetic modification techniques were developed. Recombinant DNA technology is the technique of combining two DNA sequences from different sources. One of the goals of this technique is cloning. It is a particular gene for analysis or mass production of a medically useful protein product. Bacteria, yeast, and cultured plant and animal cells are usually used for recombinant DNA experiments. The first step in recombining the DNA from different species is to clone millions of copies of single desirable piece of DNA. Molecular cloning is done by cutting the desired DNA fragment or gene into a vector, usually either from a virus or a bacterial plasmid. A small plasmid from E. coli is a circular DNA containing about 1,000 to 20,000 base pairs that can replicate independently inside a bacterial cell. Specialized enzymes are also used to cut DNA molecules in specific regions to allow the insertion of the new DNA. The process for genetic engineering begins with the same for any organism being modified. Firstly, it is to identify an organism that can contain a desirable gene. Secondly, extract the entire DNA from the organism. Thirdly, remove this gene from the rest of the DNA. One way to do this is by using a restriction enzyme. These enzymes search for specific nucleotide sequences where they will cut the DNA by breaking the bonds at this location. Lastly, insert the new gene to an existing organism's DNA. This may be achieved through a number of different processes. Genetically modified organisms or GMO have many uses in the fields of agriculture and food industry, even in medicine, research, and environmental remediation. Genetically modified bacteria are the most common GMOs because their simple structure permits easy manipulation of their DNA. One of the most interesting uses of genetically modified bacteria is the production of hydrocarbons, like plastics and fuels, usually only found in fossil fuels. Examples of genetically modified bacteria are the following: cyanobacteria that have been modified to produce plastic or polyethylene, and even fuel for butanol as byproducts of photosynthesis, and E. coli bacteria that have been modified to produce diesel fuel. Genetically modified foods are crop plants created for human or animal consumption using the latest genetic engineering methodologies. New genes are introduced for a variety of reasons, whether it's to grow higher yields, make crops more resistant to infection and pests, or even to infuse them with extra nutrients and vitamins. Among the GM foods successfully produced include the following: BT corn. It is a pest-resistant plant against corn-infesting larvae. A gene from the BT bacteria is added so the corn produces a protein that is poisonous to certain insects but not humans. Another example is the golden rice that have been developed by adding the genes for the synthesis of beta-carotene to the genetic material of the original rice species. The genetically modified rice is created to improve the nutritional value of rice through increased vitamin A content that is found in dark green and dark yellow vegetables such as carrots. Banana vaccines is also an example wherein a modified virus injected in sapling tree causes the bananas to contain virus proteins. Venomous cabbage is also a product of GMO in which genetic engineers combine the gene of poison in the tail of the scorpion with a cabbage that kills caterpillars. Soy has also been genetically modified to resist herbicides. In 2007, over half of the world's soybean crop, which is 58.6%, was genetically modified. And a fish in tomatoes and strawberries, genetic engineers inject Arctic fish genes into tomatoes and strawberries to make them frost tolerant. The aim of GM animals is to improve the yields in animal breeding, genetics, and reproduction. Genetic modification in animals has allowed the production of fishes, cows, pigs, rabbits, and sheep with increased size and mass. Examples of genetically modified animals are fast-growing salmon. The biotechnology industry says it has genetically modified fish that grows at twice the normal rate, so it can get to market sooner and make more money faster. Silky spider goat milk. Genetic engineers have created goats with spider genes that create silk in their milk. Apart from increasing the silkiness of the beverage, scientists also use its spider web protein to make bulletproof vests. Less smelly cows. Modifying bacteria responsible for methane production in the cattle results in 25% less flatulent cows. Chickens with multiple legs and wings. Genetic engineers are able to create chickens with multiple legs and wings to cope with the increasing demand on chicken consumption. Also, genetic engineering has been widely used in the medicine and human health care. Insulin is a hormone used in diabetes. The insulin from human or animal is cloned in bacteria and used for the synthesis of insulin. Production of antibiotics is also a product of genetic engineering in the field of medicine. This includes penicillin, streptomycin, neomycin, etc. Production of vaccines is also an important advancement in medicine that has made. Vaccines is an important immunizing agent. The vaccines elicit antibody for virus that have been developed using this technology. These vaccines are hepatitis B, HIV, foot and mouth disease, influenza, etc. Fourth, human growth hormone. Other medicine or treatment for cancer, immune deficiency, heart attacks, etc. What are the advantages of GMOs? The possible benefits of using GMOs include resistant to bacteria or virus, pests, weeds, and droughts. Reduce the use of pesticide, which is harmful to the environment. Increase crop and provide steady food supply to consumers. Cheaper cost for consumers. Enhance the food quality with more nutritious and improved taste, texture, size, and color. Longer shelf life, food with more desirable traits, and medicinal foods that could be used as vaccines or other medicines. Also, faster growth, higher production of milk by cows, make pigs with less fat, leaner meat, make growth hormone to treat dwarfs, etc. What are some concerns regarding GMOs? Some people have expressed concerns about GE foods, such as risk to human health, unsafe to eat, creating foods that can cause an allergic reaction or that are toxic, unexpected or harmful genetic changes, genes moving from one GM plant or animal to another plant or animal that is not genetically engineered, harm to the environment and wildlife, and foods that are less nutritious. And that concludes another episode of Earth and Life Science. Did you enjoy learning? I hope you've learned a lot from genetic engineering. To assess if you have really understood our discussion, let's have a short quiz. But before having your quiz, let's have a short review of the lesson. What is DNA? Exactly. DNA is the basic hereditary unit in all organisms. What is genetic engineering? Correct. Genetic engineering is the process of modifying genes in a living organism to produce genetically modified organisms. What are the general process of genetic engineering? Certainly. First, identify an organism that contains a desirable gene. Second, extract the entire DNA from the organism. Third, remove this gene from the rest of the DNA. Last, insert the new gene to an existing organism's DNA. What are the benefits of using GMOs? Very good. Resistant to pests, weeds, and droughts. Reduce the use of pesticide. Increase crop and provide steady food supply. Cheaper cost. Enhance the food quality. Longer shelf life. And faster growth. What are the risks of using GMOs? Correct. Risk to human health. Unsafe to eat. Creating foods that can cause an allergic reaction or that are toxic, unexpected or harmful genetic changes, genes moving from one GM plant or animal to another plant or animal that is not genetically engineered, harm to the environment and wildlife, and foods that are less nutritious. You got all my questions right. I guess you are now ready for the quiz. Continue reading your notes for you to refresh your mind on the lessons we discussed. You will have your quiz after this break.
Ladies and gentlemen, let's welcome our Miss Earth Warrior 2020 from the Philippines. Hi everyone. Miss Philippines, for you, what is the biggest problem the Earth is facing today and how can we solve it?
Earth is suffering because of us humans. Climate change and global warming are the biggest problems caused by our bad acts. So I think the best solution is the practice of 4 R's. Let's reduce our carbon emission. Let's reuse for conservation. Let's recycle plastic and let's rethink of our old ways. We only have one Earth, but the Earth has millions of us. Let's love it more than it loves us. Thank you.
Let's take care of our Mother Earth. A message from DNR, DepEd in this station.
And we're back! I will be giving you a 5-item multiple choice question. Listen carefully as I'll only repeat the question twice. Choose the letter of the correct answer. Again, I'll repeat the question twice, then choose the letter of the correct answer. Are you ready? Okay, let's begin. Number 1. The process of modifying genes in a living organisms to produce genetically modified organisms is called blank. A. Selective breeding. B. Genetic engineering. C. Inbreeding. Or D. Hybridization. Again, the process of modifying genes in a living organism to produce genetically modified organisms is called blank. A. Selective breeding. B. Genetic engineering. C. Inbreeding. D. Hybridization. Number 2. It refers to a large biomolecule that contains the complete genetic information for an organism. A. DNA, B. RNA, C. Protein, or D. Enzyme? Again, it refers to a large biomolecule that contains the complete genetic information for an organism. A. DNA, B. RNA, C. Protein, or letter D. Enzyme? Number 3. Which of the following process is not applicable in genetic engineering? A. Cut out a piece of DNA from a DNA molecule. B. Insert a piece of DNA from one organism into the DNA of another organism. C. Use a restriction enzyme to cut DNA and form sticky heads. D. Read the sequences of bases in a piece of DNA. Again, which of the following process is not applicable in genetic engineering? A. Cut out a piece of DNA from a DNA molecule. B. Insert a piece of DNA from one organism into the DNA of another organism. C. Use a restriction enzyme to cut DNA and form sticky ends. D. Read the sequences of bases in a piece of DNA. Number 4. What has been the advantage of genetically modified organisms? A. Increasing the food supply. B. Less nutritious food. C. Producing clones. D. Using more pesticides. Again, what has been the advantage of genetically modified organisms? A. Increasing the food supply. B. Less nutritious food. C. Producing clones. D. Using more pesticides. Number 5. An example of genetically modified plants that is rich in beta-carotene. A. BT corn. B. Golden rice. C. Venomous cabbage, or letter D. Papaya. Again, an example of genetically modified plants that is rich in beta-carotene. A. BT corn. B. Golden rice. C. Venomous cabbage, or letter D. Papaya. We are done with another amazing episodes of Earth and Life Science. Answers for your quiz will be given on the next episode. On behalf of our scriptwriter, Caroline A. Dawang of Cabanatuan National High School and the SDO Isabela RBI production team, thank you very much for listening. I am your teacher, Ma'am Johanna R. Caro. Until next time for another day full of learning. If you have clarifications, feel free to contact your subject teacher through text or call, email, or Facebook Messenger. Again, thank you for listening. Have a good day!
Patuloy ang edukasyon para sa ating henerasyon.
Sa daan ng pagkatuto ay walang may iwan.
Kaya halina sa Radyo Eskwela!